Welcome to our channel Mootechie. That's us, your guide into the microscopic world. Today's topic is bacteriophages. We are going to take a clean, modern, and very visual journey through these fascinating entities. In this overview, we will cover their definition, their structure, the different types, and their life cycles. And don't worry, we'll be featuring smooth animations and scientific diagrams to help everything click. The areas we'll touch on include genetics, cycles, and analysis. Think of this as your primer to the universe of phages. Now, let's get introduced. What are bacteriophages? At their core, they are defined by their viral nature. Bacteriophages are viruses that strictly infect bacteria and sometimes archaea, using them as hosts for replication. This means they can't infect human or animal cells. They are specialized hunters of microbes. A key feature of this relationship is host specificity, which is typically very high. A phage will often only attach to one bacterial strain, making it a precise tool. Their first remarkable trait is their global abundance. They are considered the most abundant biological entities on Earth, thriving everywhere from oceans and soil to the human microbiome, inside us. Next, let's consider their structure and genome. They are incredibly diverse, with genetic material that can be DNA or RNA, encased in protein coats or capsids that come in shapes like the geometric icosahedral, long and thin filamentous, or the classic head-tail design. Their scientific impact is profound. They are critical for emerging treatments like phage therapy, for biocontrol in agriculture, and as molecular tools. In fact, CRISPR gene editing technology has its origins in phage research. To grasp their scale, the estimated population of phages on Earth is a staggering 10 to the 31st power. That's a 1 with 31 zeros after it. Let's build a deeper understanding. A bacteriophage is simply put, a virus that parasitizes a bacterium to replicate. It's a foreign invader that takes over the cell's machinery. Its infectivity is often host-specific, achieved through precise receptor recognition. It's like a key fitting into a very specific lock on the bacterial surface. Phages typically employ one of two main infection strategies: the virulent or lytic cycle, which destroys the host, and the temperate or lysogenic cycle, which can be a longer dormant relationship. Ecologically, they play vital roles. They act as natural population controls for bacteria and are major drivers of horizontal gene transfer, moving genetic material between bacteria, which can spread traits like antibiotic resistance. Let's examine their anatomy in detail. Here is the structure of a typical tailed bacteriophage. Starting at the top, we have the head or capsid. This is a polyhedral, often icosahedral, protein shell that acts as a protective vault for the viral genome, which can be DNA or RNA. Below that is the collar and the tail sheath. The sheath is a contractile mechanism that acts like a molecular syringe. When triggered, it contracts to forcefully inject the genetic material into the host cell. At the very bottom, we find the base plate and tail fibers. These are the sensory and attachment apparatus. The fibers are like landing legs that scan for and adsorb or bind to specific receptors on the bacterial surface, ensuring the phage docks correctly. Regarding their envelope status, most bacteriophages are non-enveloped, meaning they don't have a lipid membrane stolen from a previous host. Their complex morphology is directly tied to their infection mechanics. Every part has a job in the invasion process. Phages come in different forms, so let's classify them. The main morphological types, often under the order Podoviridae, are defined by their tails. First, the Myoviridae family. These have a complex contractile tail with a sheath that shortens during infection. A classic example is the well-studied T4 phage. It mechanically injects its DNA like a hypodermic needle. Next is the Siphoviridae family. These have a long, flexible, non-contractile tail structure. An example is the lambda phage, famous for its lysogenic cycle. This is actually the most abundant morphology found in nature. Finally, the Podoviridae family. These have a short, stubby, non-contractile tail. An example is the T7 phage. It often uses a simplified injection mechanism that relies more on enzymes to get its genome into the cell. It's also important to note that phages can be classified by their life strategy, virulent versus temperate, and by their genome type, which can be double-stranded DNA, single-stranded DNA, or even RNA. Now, let's explore viral replication starting with the destructive lytic cycle. We can break it down into 5 clear stages. First, attachment, or also called adsorption. This is where the phage tail fibers bind to specific surface receptors on the bacterial cell wall. It's the critical first step of recognition and docking. Second, penetration. The tail sheath contracts, driving the tail tube through the cell envelope and injecting the viral DNA or RNA. The empty protein capsid remains outside the cell. Third, replication or biosynthesis. Here the phage hijacks the host cell's machinery. The bacterial cell is forced to stop its own work and instead synthesizes new viral genomes and the proteins needed to build new phage particles. Fourth, assembly or maturation. This is where the new viral components self-assemble. The heads are packed with the freshly copied DNA and the tails are attached. Complete infectious phages form inside the doomed cell. And fifth and finally, lysis or release. Phage enzymes called endolysins are produced, which rupture or degrade the bacterial cell wall from within. The cell bursts open, or lysis, releasing typically between 50 to 200 new phage particles, now ready to go and infect neighboring bacteria. But not all infections are immediately destructive. Let's look at the alternative, the lysogenic cycle, a state of viral dormancy. This cycle has about 6 stages. It begins the same way, attachment and injection, where the phage binds and injects its DNA. The key divergence happens next. Instead of taking over, the phage DNA undergoes integration. It recombines into the host bacterium's own chromosome, becoming a hidden passenger called a prophage. Then, during normal cell division, the bacteria reproduces. The prophage DNA is passively copied along with the host DNA and passed on to all the daughter cells. This creates a population of lysogenic bacteria or lysogens, and the prophage is maintained in a state of dormancy. This is managed by repressor proteins that prevent the expression of the lytic cycle genes, keeping the phage DNA silent. This can last for many generations. However, the prophage can be activated by an induction trigger. Environmental stressors like UV light, chemicals, or starvation that damage the bacterial DNA can inactivate the repressor proteins. This triggers the final stage, excision, leading to the lytic cycle. The prophage DNA cuts itself out of the host genome and then switches gears to enter the full lytic cycle we just discussed, replicating and ultimately lysing the cell. Thank you for watching. We've been exploring the invisible world with Mootechie. Let's do a quick session recap. We started with the definition of what a bacteriophage is. We then broke down its intricate structure. We categorized the main types based on their morphology, and we walked through their 2 primary life cycles, the lytic and the lysogenic. We hope this foundation gives you a clear picture of these tiny but powerful entities. Stay connected with us and please subscribe to Mootechie so you don't miss our upcoming microbiology deep dives.